Hi everyone, welcome to our Biostatistics tutorial series. In this lesson, we'll be diving deep into a core concept in every scientific experiment called variables. Whether you're conducting a lab experiment, running a survey, or just reading scientific papers, you will keep seeing this word pop up. So, what exactly are variables and why are they so important? Let us find out. What are variables, you may want to ask? Variables are characteristics or quantities that we can measure, observe, or categorize. They are called variables because they can vary, that means they can change, and they can take on different values in different people, groups, or over time. For example, someone's blood pressure, age, or even the number of hours they sleep per night are all variables because these values change from one person to another. In research, we use variables to help us understand what is happening. We measure or manipulate variables so we can explain relationships, make predictions, or test ideas. And for example, if you want to know whether exercise reduces stress, you will look at how changes in exercise level affect stress levels. Those are your variables. So let's talk about the most important three types of variables you need to know. Number one, we have independent variable. Two, we have dependent variable. And thirdly, we have control variables. Each of these plays a unique role in the research process. Think of them as characters in the story. The independent variable is the one you think causes something to happen, while the dependent variable is the effect or the outcome of that thing which is to happen. And the control variables are the background settings you want to keep the same so that they don't interfere with your story. Let's go deeper and take a look at what independent variables mean. So, an independent variable is the one that you change, manipulate, or observe to see its effect on something else. In experimental studies, researchers will actively change the independent variable. Meanwhile, in non-experimental or observational studies, they simply observe it without manipulating it. Here are a few examples of independent variables. Number one, let's say in the study on the relationship between air pollution and asthma in children, our independent variable in this study will be air pollution, which can be measured but not changed by the researcher, by the way. And our dependent variable will be asthma cases, which will be the outcome. Please, let's take a look at it again. In a study on the relationship between air pollution and asthma in children, air pollution is our independent variable because it is the one causing an effect on the rate of asthma in children. Then that means asthma is going to be our dependent variable. It is depending on the air pollution, so it is the outcome. Let us look at another example. In the study of the effect of different doses of vitamin D on calcium absorption, in this case, our independent variable is going to be vitamin D dose, which can be manipulated by the researcher. The researcher might decide to increase the dose of vitamin D or lower it. So, increasing it or lowering the dose is going to have varying effects on the rate of calcium absorption. So, our independent variable here is the vitamin D dose. Then, our dependent variable will be the calcium absorption. In a survey of the effect of the time spent on social media and student concentration in class, our independent variable here is going to be social media usage or time spent on social media. Then our dependent variable will be concentration levels, which means if students spend more time on social media, it's going to have effect on their concentration levels. And if students spend fewer time on social media, it's still going to have a different effect on their concentration level. So the concentration levels in class is depending on the time spent on social media. In all of this, the independent variable is the input. It is what you think might be causing a change or an effect. So now let us take a look at dependent variables. The dependent variable is the output. So please, let's get that straight. If the independent variable is the input, the dependent variable is the output or the outcome. It is what you are measuring as the result or effect, and it depends on the independent variable. In short, it is the outcome you are trying to study. Let us revisit our previous examples. If you think that vitamin D helps with calcium absorption, the absorption is what you measure. It is your dependent variable. And if you think that more time on social media reduces focus and concentration, then the concentration levels are what you measure. They are your dependent variables. So the key idea here is independent variable causes and then dependent variable response. Now let's take a look at the third type of variable, which is called control variables. These are variables that could affect your results, but you don't want them to affect your results, so you keep them constant. You keep them the same. Why? Because if they change, they might affect and confuse your results. Let's take this example. In this question, does caffeine improve running speed? Does caffeine improve running speed? Caffeine intake is going to be our independent variable, while running speed will be our dependent variable. Then our control variable in this sentence or in this question is going to be different factors such as age, body weight, or even the type of running surface and type of running shoes. Why do we need controls? In this research question, we need to keep controls because younger and lighter runners may naturally run faster than the older ones. So if you don't keep control for that, we won't know if the speed increase was from caffeine intake or just because the runner was young and athletic. Let's now discuss what we call positive and negative controls. In experiments, we talk about positive and negative controls. That's because they help us to check that our experimental setup is working correctly. Let's break it down. A positive control uses a treatment that is already known to work, and it should produce the expected results. For example, in a bacterial antibiotic sensitivity test, a known active antibiotic is added and expected to kill the bacteria. If the bacteria dies, we say our positive control works. But if the bacteria doesn't die, then there may be a problem with the test. Also, a negative control uses no treatment at all. It shows what would happen naturally without interference. For example, in the same bacterial antibiotic sensitivity test, if you don't add any antibiotic, then the bacteria is not expected to die. But then, if the bacteria still die, then something is wrong. Maybe the culture plate might be contaminated. So, let's look at two summary examples that combine everything we've learned in this class. Number 1. A study tests the effect of light intensity on plant growth, keeping water and soil constant. In this example, our independent variable is going to be light intensity. Our dependent variable will be plant growth. Our control variables will be water and soil. Then our controls is going to be positive controls that is the plant growing under optimal light. In the COVID-19 vaccine trial, one group gets the real vaccine and another gets just salt and water. In this study, our independent variable is going to be COVID-19 vaccine. Our dependent variable will be immune response. Then our control variables will be H and S status. And we can identify both positive and negative controls in this study. Our positive control will be the real vaccine group or the real vaccine, while our negative control will be salt and water. This example just show how researchers test real-world problems while keeping their experiment clean and trustworthy. So, let's test your understanding with a quick exercise. You should read this scenario and answer the question. Researchers want to know if using face masks reduces the spread of flu in a secondary school. Question 1. What is the independent variable? Question 2. What is the dependent variable? Question 3. Name at least 2 control variables in this study. Question 4. What could serve as a positive and negative control? Pause the video and think about it, and then drop your answers in the comment section. So to summarize this class, let's say variables are the building blocks of all research. The independent variable is what to change or observe, and the dependent variable is what to measure, and control variables are what to keep constant. Then remember, a good experiment always includes both positive and negative controls to keep our work reliable. In the next lesson, we are going to explore types of data and measurement scales like nominal, ordinal, interval, and ratio data. Don't miss it. And if this video helped you, please like, share, and subscribe. See you in the next episode.